Okay, for chapter 9, we've been talking about the three domains: domain bacteria, archaea, and eukarya. Let's now focus just a little bit more on domain bacteria. So there are producers, consumers, and decomposers in the domain bacteria. And what that means is that there are heterotrophs and autotrophs. A heterotroph is something that consumes another organism in order to gain nutrients. So that includes things like consumers and decomposers. Some heterotrophs are photoheterotrophs, meaning that what they consume is primarily what they make themselves via sunlight, but they can also eat other organisms. Or chemoheterotrophs that primarily absorb chemicals from their surroundings or their environment, but can also eat other organisms. For example, saprophytes feed on dead organic matter. They're primarily after the chemicals, but they're consuming something that comes from another organism. Autotrophs are also known as producers. There's photoautotrophs and chemoautotrophs. A great example of photoautotrophs are cyanobacteria, which are bacteria that contain chloroplasts or light absorbing organelles, and that's going to be utilized in order to carry out processes like photosynthesis and produce oxygen. Chemoautotrophs can do something similar, just not with sunlight. They do it instead with chemicals in their environment. So, speaking of using processes like photosynthesis in order to make oxygen, we can also talk about processes that occur in oxygenated environments, like cellular respiration. Most bacteria actually perform aerobic cellular respiration, aerobic meaning with oxygen, whereas the term anaerobic would mean without oxygen. Something called an obligate aerobe would require an almost constant supply of oxygen, whereas an obligate anaerobe, they could carry on their metabolic processes without oxygen. And then facultative anaerobes use oxygen if it is available, but they do not require it. Some bacteria also form endospores, especially when their growing conditions become unfavorable. Endospores are protective. They are a thick internal wall, generally made up of several hard fibrous layers, and they can be produced by bacterium. They generally enclose the important information, like its genome or DNA, and other essential parts, like ribosomes, used to make protein from that DNA blueprint. Endospores will actually allow bacteria to stay dormant in extreme conditions for months or years until it is safe to begin reproducing again. So, speaking of bacterial reproduction, not only do they reproduce in unique ways, they also can grow if they are in the correct environment. There are many conditions that must be present for bacterial growth, including moisture, a moderate temperature, nutrition, darkness, now there are some exceptions there, and the proper amount of oxygen. Moisture is required for bacterial growth and reproduction. Whether it's through capsules or the use of endospores, we can see bacteria utilizing its environment only when it has the right amount of moisture. In very desiccated, dry environments, it will protect itself. It needs to protect itself from drying conditions. Now, summer would be the most ideal season for bacterial growth. We need a certain amount of heat, but extreme heat can kill bacteria. Nutrition varies, and darkness can also vary among different types of bacteria. Many prefer darkness because of the possibility of damaging UV radiation. And oxygen is required from facultative aerobes as well as obligate aerobes. But bacteria only reproduce asexually. They use a process called binary fission, similar to the mitosis of eukaryotic cells, but binary fission is a little simpler. Now, we also see with binary fission a higher amount of mutations. They can occur during this process just like they can in mitosis or even meiosis. There are plenty of processes of genetic recombination in bacteria that can also produce modifications of the DNA. One is transformation, and that's when the transfer of a DNA fragment from a non-functional donor cell to that of a functional recipient cell. Or transduction, the process in which infection from a virus results in DNA being transferred from one bacterium to another. Or we could say conjugation, the temporary union of two organisms for the purpose of DNA transfer. So conjugation can occur when we see exchange of material between bacteria. Oftentimes they'll exchange DNA in the form of a plasmid. That can give them a special function. Maybe, maybe not, but that's something a plasmid could code for. With transduction, we see something called a phage, generally a bacteriophage, carry bacterial DNA between cells. There are viruses that affect bacteria, and they generally allow for the process or function in the process of transduction. And with transformation, generally we'll see the release of genetic material from a damaged bacterial cell. Because bacteria can take up material in their environment, the recipient cell can take in foreign DNA and then incorporate it into their own plasmid or their own genome. Again, that's transformation. So here is a pictograph showing binary fission. You can see the base processes are prokaryotic cells like bacteria growing by increasing in cell number, and that's probably going to be most likely not because the cell is lengthening before it splits. We see the biggest increase in cell growth by the actual copying or splitting of the cytoplasm to create two new cells. That replication by binary fission, which is the splitting of one cell into two, can have a profound effect on bacterial populations, and we definitely see that bacterial populations tend to increase by a factor of 2 or double every generational time. Here we see an example of transformation. We see DNA fragments from donor cells, possibly dead bacteria, floating in the environment of a living bacteria cell. This recipient cell can take up the donor DNA, and recombination can occur when that donor DNA is incorporated into the recipient DNA or into the genome of the cell, and that would create a genetically transformed bacterial cell. Now, these processes are occurring, or what it seems like, are occurring in bacteria cells pretty easily. They do not occur in eukaryotic cells to the same rate, if at all, because of the complexity of eukaryotic cell DNA structure and the fact that that DNA is sequestered away or within a nucleus. Here we see a pictograph of transduction. We see that the DNA of a phage, so a bacteriophage, penetrates the bacterial cell. The phage DNA may become integrated with the host cell DNA as a prophage, so that phage DNA can be incorporated into the genome. And when the prophage becomes lytic, meaning the virus material is trying to copy itself but then escape the cell, we would see that the DNA genome to which it was inserted is degraded, and all the other components of the new phages, including their genome, are reproduced or copied, creating more bacteriophages, or essentially more structures that contain viruses that can infect bacteria. And that means that this infected bacterium, even though it's taken part in transduction, is actually now going to be destroyed. And this is one example of transduction that actually ends in the lytic cycle, or the busting of the bacterial cell to release bacteria. And then that would allow these new phages that were produced in this infected bacterial cell to go infect new host cells and introduce their genes into the host cell. And this would continue on. We start to see kind of the buildup of genomic recombination within bacteria. Conjugation is when the donor cell is going to send information or genetic information to the recipient cell, but via a connection. So we see a donor cell attached to a recipient cell with a pilus. A conjugation pilus is going to hold the cells in contact with each other. And then we see one strand of plasmid DNA transfer to the recipient. Plasmids are small pieces of genomic information that can code for special abilities like antibiotic resistance, but don't always. And we can see also that the recipient synthesizes a complementary strand to these piece of plasmid DNA they have so they can complete that replication, and so can the donor cell. They both can restore the complete plasmid structure. Now, when we're looking at bacteria, you probably think of disease. And when we're talking about bacteria and disease, we have to make sure we mention that not all bacteria cause disease. Some bacteria, like probiotics, are good for us. They live in our intestines, they help us make vitamins. Others are pathogenous, and the term pathogen means something that is disease causing. And pathogenic bacteria therefore cause disease, and generally they do so by either damaging cells and tissue or producing toxins. They can produce exotoxins, poisonous proteins produced as a part of bacterial growth and metabolism that cause damage to cells and tissues that the bacterium has invaded, or endotoxins, which are poisonous lipids found on the bacterial cell wall. And this type of ligand is an integral part of the bacterium, is released when the bacterium dies and the cell wall starts to break up. So different types of toxins, some are produced or secreted, exotoxins. Others are made within and only released upon the death of the bacterium, endotoxins. So if we're talking about human disease, especially against bacterial infection, we have to mention the lines of defense that the human body has. We have the first line of defense, which is our surface barriers, our skin, and our mucous membranes. We have the second line of defense, all of the internal chemical and cellular attacks, including cells like neutrophils or macrophages that can, if they come into contact with the bacterium, eat it up and destroy it. And then we have the third line of defense, which are targeted protein and cell attacks, which use very special protein and pathways in order to go after only a particular type of pathogen. In order to help our body fight off disease, we can prevent essentially pathogens from getting into our body, at least to a certain extent. So, washing our hands, maintaining good hygiene, they are all great preventative measures for spreading bacteria. Adequate amounts of sleep and good nutrition are also important. They help our immune system stay strong. So, thoroughly cleaning and cooking food, maintaining good hygiene, washing our hands, all of this can prevent bacterial contamination of what we touch, consume, and of that in our environment. And you also want to make sure that you're dehydrating or freezing foods. And that's always a good option if you're trying to do some type of preventative measure in order to make sure that bacterial growth cannot take place. Next, we're going to briefly speak on viruses. So, viruses are smaller than bacteria. In biology, we do not consider viruses living. It's non-cellular, but it is infectious, and it generally has two characteristics. It has genetic material. Can be DNA, might be RNA, and this genetic material will be inside a protective protein coat. Not a nucleus, just a protective protein coat. And therefore, it cannot reproduce on its own. It's basically DNA and protein coat. And so to use replication machinery, it has to infect a cell, whether that be a prokaryotic cell like a bacterium or eukaryotic cells like those that we're made up of. So again, the basic viral structure, genetic material, and a protein coat. This protein coat is generally called a capsid. So here we see some examples of viruses. This is the tobacco mosaic virus, and you can see that it has a spiral capsid and an RNA genome. And the genome is inside the capsid. This spiral capsid or protein coat is generally elongated to create a rod-shaped virus. Here we see an adenovirus. We can see that its genome is DNA, and that's inside a protein coat or capsid. And it also has antenna-like fibers that make up part of its capsid. So, based on the general description of a virus, you should have picked up on that it's not alive. It doesn't have all of the organelles or structures that allow it to reproduce itself. And being able to reproduce is a primary characteristic of living organisms. So viruses fail to meet one of life's criteria. But there's many different types of viruses, regardless of their status as non-living. There's bacteriophages, DNA viruses, and RNA viruses. So here is a better look at a bacteriophage. This is a T4 bacteriophage that can infect T helper lymphocytes. It does kind of look creepy. It does have tail fibers that generally anchor it to the T4 cell. It does have a tail that it can squeeze its DNA out of, and it has an upper hexagonal capsid or protein coat. This type of virus, a bacteriophage, infects bacteria. So here are multiple phages infecting E. coli bacterium. Keep in mind, viruses are not cells. They cannot metabolize independently. They are going to be using infected host cells to even replicate their own viral DNA. So that's why we see viruses invade other cells. They have to take over that cell's machinery, whether it be for nutrients or cell translation and transcription to reproduce. Now, cell translation and transcription is generally what we see if a cell contains, or I should say a virus contains RNA. They will have to backtrack, create DNA in order to get their genetic material. Viruses tend to be very specific to the cells that they infect because they actually have to bind to the cell's precise surface proteins. And all of our cells have slightly different surface proteins. Viruses infect cells in one of two circular pathways. So we're going to look at those two circular pathways, the lytic cycle or the lysogenic cycle. The lytic cycle is when a virus enters a cell, hijacks the host cell's DNA replication system, makes copies of itself, and causes the cell to burst, releasing more viruses. So with the lytic cycle, a virus takes over, makes more of itself, but to get out, it kills the cell it infects, or busts it open. With the lysogenic cycle, a virus enters a cell, embeds its DNA into the DNA of the host cell, and is replicated along with the host cell's DNA. So with the lysogenic cycle, a virus enters, kind of hides its DNA in the genome of the host cell, and is replicated kind of without the cell possibly noticing that it's been infected. This makes the host, if it's made up of many cells, less likely to ring an immune alarm and alert the rest of the body to an infection. When we're looking at viral infection, we generally focus on animal cells, eukaryotic cells, those with the most complicated surface attachment proteins, and we still see that there are viruses that can bind to these specific cell receptors. They can fuse with the plasma membrane, and then sometimes be taken in via endocytosis. An invagination process via the plasma membrane forms a vesicle around the infecting virus and takes it inside the cell. That's going to be more possible if we have a good fit for the surface proteins on the capsid of the virus and to those receptors on our eukaryotic cells. So here we see the membrane fusion that takes place via the process of viral infection. We see up top that the virus attaches to specific proteins. There are receptors on the plasma membrane of the host cell. Then we see membrane fusion, and the viral envelope is fusing with the plasma membrane. The virus is then released in the host cell's cytoplasm with its capsid surface proteins remaining in the plasma membrane. Then the viral nucleic acid, or genome, separates from the protein coat, the capsid, and that can enter the host cell's nucleus and replicate. The viral nucleic acid is going to be transcribed into mRNA, and the host ribosomes are actually going to direct the translation of that mRNA to create viral proteins that create new capsid coats or protein coats. And then a vesicle will transport the glycoproteins created for new capsids to the host cell plasma cell membrane. And these components tend to come together right before the cell leaves via exocytosis, or the virus leaves via exocytosis, or right after. Now, we could also look at viral infection of plant cells. And in this case, because of the extracell wall that's very rigid and structured, we see that viruses generally cannot penetrate cell walls, unless, of course, they are damaged. So we see spread by insects that feed on plants or by infected seeds. We see that as the best process by which viral infection occurs in plants. And we see some spread throughout the plant via plasmodesmata, or those gap junction-like cells that connect plant cell to plant cell. All right, again, the lytic cycle is destroying the host cell. Lysogenic is not. Basic steps of the lytic cycle: attachment, penetration, replication, assembly, and then release. The release is going to be destructive for the host cell. So notice we have attachment, penetration, replication, creating more DNA or RNA, more protein coat. Once we put them together, in order for the newly made bacteriophages here to get out, they must be ejected, and they're only going to be ejected via cell lysis, which is going to destroy the host cell. Lysogenic cycle usually doesn't kill the host cell. That doesn't mean it can't, but its goal is not to kill the host cell. Generally, it's to go kind of unseen. They actually can have a dormant stage for many lysogenic bacteria. Herpesviruses that cause chickenpox and cold sores are generally found using the lysogenic cycle. And viral genome replication is going to be with insertion of viral DNA, whether into bacteria or eukaryotic host cell DNA. So we're going to see basic steps: attachment, penetration, integration, and replication. So here we see attachment of a bacteriophage, penetration of genomic sequence, then insertion or integration of that genomic sequence into either a bacterial host or it could be another animal cell host, and then replication, meaning that genomic sequence during a dormant cycle will remain in the genome of the bacterium, pretty much be hiding in that genome, but then when certain signals occur, that genomic sequence can be replicated to produce more bacteria that can be secreted or escape the cell. Now we also have to talk about retrovirus reproduction cycles, because while most viruses contain DNA, there are RNA viruses, and they would need other enzymes such as integrase in order to integrate their DNA into the host chromosome. So they would need to turn their RNA genome into DNA before they integrate it. So we would see that viral DNA used to transcribe viral RNA and synthesize proteins are generally going to be present in the host cells of those viruses that do contain RNA. We can also look at some examples of viral infections like warts, chickenpox, the common cold, influenza or the flu, some forms of cancer, mumps, measles, and AIDS. So those are just some examples of viral infections. And the best defense against viral infection, again, is prevention, good hygiene. Washing your hands, making sure that you put your food away in a hygienic manner, whether you are freezing it or dehydrating it. And we do have an immune system. The human immune system can fight off viral infections. They use both phagocytic white blood cells and also targeted chemical messaging. Our immune system does work best if it's seen a virus before, but the production of antibodies will occur regardless in response to viral infection. We also see immune system activity with bacterial infection, but you may not see antibody production with bacterial infection. But generally, we see some type of immune response that helps to destroy a pathogen. Our body can even create memory cells. And memory cells for an infection, viral or bacteria, are primarily made after a first infection and help a second infection by the same virus or bacteria help us treat it a lot faster. Now, vaccines are supposed to mimic primary infections, and that way we do not go a prolonged period of time before our really focused immune system is able to act against a virus or a bacteria that our body has never seen before, because secondary responses are always faster and stronger than a primary response. So, tricking our body into thinking we've seen a pathogen before is the idea behind vaccines. Vaccines contain a weakened or inactive version of a pathogen that stimulates the body's production of antibodies that aid in destroying the pathogen. Vaccines are given to protect or aid against virus infection, not bacterial infection. And we can also talk about an interesting case, the first vaccine development. It was actually made against the smallpox virus. It was made by English physician Edward Jenner in the late 1700s. And back then, many people came down with smallpox. Some died. And even if they didn't die, it was a disfiguring and very damaging disease. And he observed that milkmaids at that time in the 1700s, when they became infected with cowpox, which was basically a milder disease than smallpox, but similar, they actually never contracted smallpox. So, what he hypothesized was that, well, cowpox and smallpox viruses must be so similar that the immune system of the milkmaids was being fooled into defending against the body or defending the body against smallpox, even though the milkmaids had never had the disease. Basically, cowpox and smallpox were so similar, at least in how the virus looked, that when milkmaids came into contact with smallpox, they didn't have symptoms because their body mounted a secondary response or had been fooled into thinking it had seen this before. And they were able to fight off a disease that could be disfiguring and fatal, whereas others were not. Now, Jenner came up with a pretty famous experiment, but definitely very controversial. He used fluid from the sore of a milkmaid that had cowpox, and he inserted that into a scratch he had made on the arm of a young farm boy. Now, later he exposed that same boy, who he had exposed to fluid from cowpox, he exposed that boy to smallpox, and the boy never became ill with the smallpox disease. So that supported Jenner's conclusion that inoculation with the cowpox virus causes the immune system to recognize the smallpox virus, making a stronger secondary action be what occurs versus a primary reaction, which is slower when the individual did contract smallpox. So it thereby initiated a substantially quicker and stronger immune attack on the smallpox virus and prevented disease. He was so confident in his conclusion that he actually vaccinated his own child shortly thereafter. Now, vaccines have been made against many viral diseases, including polio, measles, the mumps. However, rapidly mutating viruses like human immunodeficiency virus or HIV and the common cold are nearly impossible to treat with vaccines. And that's just because they have to be specifically designed against singular pathogens, against a certain strain, because of how quickly some viruses do mutate. Some of them mutate so quickly they look completely different, and a vaccine against one strain would not protect against another. And that is it for chapter 9, and we're going to start chapter 10 next class. Make sure to review this chapter and prepare for the next unit.